Thank you so much. Yeah, welcome again, and thank you for the opportunity to speak at this conference about metabolomic profiling of cultured yeast and spontaneously fermented coffees. Here you can see roughly what I'm going to talk about in the next 15 minutes. And I'm going to start with the motivation and why I am doing what I'm doing. And that is my PhD project, which is a BMBF-funded project called Deep Autent. And it concerns itself with the development of benchtop non-target screening approaches for food and foodstuffs, which should be usable at the point of care. And in the end, there should be a AI-based authentication method for especially lemon and coffee. And as you might have guessed, I'm looking into the coffee field, and especially into coffee cherry fermentation. And also, I guess most of you know that there are many entry points during the coffee processing chain where microorganisms can potentially go to the coffee cherry or to the already pulped coffee and can cause off flavors. And Dr. Schwartz developed a very simple fermentation process to get rid of those off flavors, so for flavor enhancement of coffees. And the challenge that we saw in the working group is the authentication of this fermented coffee at the point of need, so that, for example, a roaster can fastly screen their coffee to see whether this coffee was really fermented with the microorganism that it is claimed to be fermented with. The challenges that we saw here is that up until now, the state-of-the-art techniques are mostly benchtop techniques that are confined to a lab. We don't want to replace those techniques, but we found that GC-IMS is a technique that is operated with nitrogen as a carrier gas and especially works at ambient pressure. So it is possible to operate this instrument, for example, from the back of your trunk, exactly where you need it. And then screen your samples, and afterwards go into the lab, and with the help of enrichment techniques as well as GC-IMS, you can then further authentify your sample. How does this GC-IMS work? We are sampling the headspace above the sample, so we are only roughly milling the sample, and we are hoping that we also can do this without milling the sample. So just adding the bean into this vial, then incubating it at a specific temperature, and then sampling the gas phase above this sample. We subject that to gas chromatography to separate the compounds inside of the sample by their boiling points. And afterwards, the compounds are then ionized in the IMS cell by a very soft ionization, and then separated by drifting alongside an electrical field against a gas stream of nitrogen. And as you can maybe imagine with the nitrogen molecules inside of this little tube, that molecules that have a very small size are drifting faster than molecules that have a very large size. And this is called the so-called collisional cross-section. And by this, we get a second separation dimension in our data. We also have a very high sensitivity in this instrument, so we can measure down to a low micrograms per kilograms concentration and generate highly complex 2D data with it, which we can optimally use for non-target screening. Now, I'm trying to explain non-target screening. I know that there are some non-target screening experts in the auditorium, so forgive me if it's not all right. So non-target screening is, the definition is hypothesis-free analysis of substances, of all substances in a given sample. And if you know what targeted means, that you have a look at specific substances or compounds in your sample, then this is the exact opposite. So we are more or less having a look at, as we are looking at the volatiles in our sample, at the whole fingerprint that those volatiles are. And this is called the volatilome. So again, you can see here that we have an instrument that has very low maintenance and operation costs, and that works under ambient pressure. However, we have very little substance-specific identification measures for this because it is not mass spectrometry. The question that we still asked ourselves was whether we can differentiate with this volatile fingerprint the different fermented coffees with this GC-IMS via the gas phase. So we tried to combine both of best worlds by hyphenating one of our IMS cells to an already established GC-IMS system. And now those of you who know MS are maybe a little bit confused because on the right-hand side you see our QMS spectrum. However, it is depicted not as a usual MS spectrum, but in this 2D plot where you have the mass on the y-axis and the retention time on the mass on the x-axis and the retention time on the y-axis, just to make it comparable to what we see in the IMS spectrum. With this, we can utilize the best part of both, so we get the soft ionization from the IMS cell, as well as the intact molecules by that, or intact ions, while with the IMS we can go to, for example, a NIST database to have substance identification. Now let's come to our method for this GC-IMS workflow. I used a Cultured-Yeast vermelho, so no species but variety. Species would be arabica, I think. And I fermented this in Brazil at Fazendas Dutra without the addition of yeast and with the addition of 3 different yeasts each. They were then subjected to an identical drying, dehulling, and roasting process, and then I analyzed them in the GC-IMS instrument, and then used the data that came out there for a non-targeted data evaluation with Python, with our in-house Python tools that we developed. I'm not trying to go into too much detail because I know this can be quite overwhelming. However, after we measure the samples, we get a raw data, so an IMS spectrum in this case, and after some alignment, spinning, and cutting, we are then left with pre-processed data, which we can then use in chemometric models, such as, for example, a PCA, which is an untargeted model, a so-called exploratory model, where we are just having a look at the differences that our samples have without having a look at the labels. So we are just putting all our samples into the model. We are not telling the model which sample has which label. And then we see how those samples separate. And then afterwards, after the model has done this separation, or non-separation, we are then having a look at the labels and see whether the separation is consistent with our labels. On the other hand, we have a PLSDA, so a supervised chemometric model, where we then add the labels. So we give the model the labels of the samples as well as our data of the samples, and then the model tries to differentiate or find the most differences between those labeled data. So overall, to maybe summarize this a little, in PCA, we maximize the variance between samples for a cluster analysis without taking into account the labels, while in PLSDA, we maximize the covariance between variables for classification, taking into account the labels of the sample. We have some limitations in this system of trying to compare GC-IMS and IMS in the same instrument, which is that while we are able to at least preliminarily assign some compounds to the GC-IMS or in the GC-IMS data, it is more or less impossible to find the compounds again in the IMS data. That is just because the IMS is more sensitive than a QMS system, so we get more compounds resolved in the IMS, and that is just such a huge mass of data that we are not able to go back to the IMS and say, okay, this compound is this compound in the MS spectrum. Just to maybe stump you on that, the MS spectrum would be the white line on the right-hand side of the spectrum. And because we have this limitation, one idea was to go to high-resolution mass spectrometry to do a more in-depth metabolomics and also volatilomics workflow. Up until now, we only established the metabolomics workflow with an LC coupled to an Orbitrap system. However, we plan also to couple a GC to this Orbitrap system to get more in-depth information also about the volatiles, and then subject this to an untargeted workflow. So for this, I used the same samples again, but I extracted the samples for LC-use and pooled always 10 of the samples per class or per fermentation, and then used an untarget metabolomics workflow. Now coming to the results of my work that I tried to explain to you here a little bit. Here you can see a PLSDA, so a supervised model of the GC-IMS data, where you can see that this red dotted line, which is a threshold, and every dot that is above this threshold is classified to a specific fermentation. In this case, on the left-hand side, you can see the wild fermentation. So every dot that is above this red line is classified as a wild fermentation, so without the addition of yeast. That worked very well. So we have a high classification precision, as well as that the majority of our samples were classified correctly. And the other part of this work that I already wanted to show you some preliminary results to is the LC high-resolution mass spectrometry analysis, where we try to go the next step to a deeper understanding here. As you can see, there are 223 detected features, features being the compounds or ions that were detected. However, this is everything that is that was detected inside of the coffee samples. So the problem that we saw was that if we only have a look at one single compound that has a molecular formula assigned to it, then we can get up to 11,000 or more results for a compound because of structural isomers. So what we are doing, instead of having a look at all of the compounds, we are trying to go for exploratory data analysis. So we are first having a look whether our samples are separating in a PCA, you remember, without the labels, just having a look at the differences between our data. And here we can see that we have some putative clusters, so we definitely need much more data to see those or to have those clusters really have meaning, but we see that there is a trend in clustering of those samples. Furthermore, I did a ANOVA. I didn't explain that, I know, but this is only useful for me at the moment to have a look at those volcano plots, the so-called, and you can see that there is this down-regulated and up-regulated over those red and green boxes. And what we are doing here is we are comparing two fermentation, one without addition of a microorganism and one with the addition of a yeast, and then have a look in which compounds are specifically found in a higher amount in the wild fermented coffee in comparison to the compounds that were found in the specific yeast fermented coffee. And the down-regulated here would be the ones that are found mostly in the wild ferment, and the up-regulated that are found mostly in the yeast fermented samples. So we are trying to just analyze those compounds to then really see the difference between the fermentations. And as you can see here, there are at least some substances that should differ or that show a difference between our fermentations. So I'm already coming to my conclusion, which is that it is possible to differentiate between cultured yeast and spontaneously fermented coffee in general, but also it is possible to classify the yeasts that were used in general to ferment this coffee. I also could show you some promising first tentative results of the metabolomics research that I did, and in future we definitely have to do a further method optimization for the GC system as well as also for the LC data analysis method, and also we try to implement the GC high-resolution MS and measure more samples in our high-resolution system. With that, I'd like to thank the cooperation partners for funding my project, my working group for always supporting me, as well as Dr. Schwartz for bringing me to Brazil, and the guys in Brazil for being so nice and yeah, making this a really special stay. Thank you so much.